Hyperplasia is a phenomenon occurring when the number of tissue cells increases in response to damage. The first thing to say is that this phenomenon arises in conjunction with hypertrophy, but only in the tissues made up of replicating cells. In fact, cells may be classified into 3 categories on the basis of their ability to replicate. This classification is called Pizzocero classification. The labile cells are those that multiply constantly throughout their life, and this is the case of the inner and outer layer epithelial cells, which are endlessly cycling cells. The stable cells instead are normally in the G0 phase, but they can enter the G1 phase when needed. Another interesting example is given by the liver cells that, in case of damage, restart the cell cycle pathway to repair it. The endothelium and the excretory ducts of the glands do the same. Then, there are the perennial cells not replicating or replicating sporadically, which live constantly in the G0 phase. Among the perennial cells, we can find myocardiocytes, striated skeletal muscle cells, and neurons, although it has been shown that in exceptional circumstances, these cells may replicate. So hyperplasia can be classified into different ways, among which there are physiological and pathological hyperplasia. A well-known form of physiological hyperplasia is that of the uterus during pregnancy, which is associated with smooth muscle cells hyperplasia. Another example is that of breast growth during pregnancy and puberty due to the hyperplasia of the gland cells. Besides the physiological hormonal hyperplasia, there is another form of hyperplasia which is compensatory and physiological as well. The most known example is that of the liver, which contains hepatocytes that are basically in a quiescent state, but that in case of damage, they may join the cell cycle and replicate to compensate the damage, after which this form of hyperplasia is named compensatory hyperplasia. The liver is in fact an organ with a remarkable regeneration capacity. The myth of Prometheus fails to store a titan that as a punishment was tied to a rock and obliged to have his liver eaten every day by a giant eagle. But Prometheus' liver regenerated in 24 hours before being eaten by the eagle the next day. Well, in real life, this phenomenon can be observed in the so-called partial hepatectomy, during which a piece of the liver is removed. Although the original shape cannot be restored, the liver mass completely regenerates after several weeks. Apparently, this form of hyperplasia could be applied to other fields. In the event of a partial hepatectomy, the liver cells release several inflammatory signals to recall some inflammatory cells. These inflammatory cells start to produce some inflammation mediators having a stimulating effect on the liver cells. In this way, interleukin-6, which binds the IL-6R receptor, and the TNF-alpha, which binds the TNFR1, is produced. These two receptors generate a signal inducing an increase in the expression of the CMYC replicating proteins and the C-focus C-Jun dimer. These genes determine the hepatocyte transition to the G1 phase, and this response is called immediate early response. To top it off, ITO cells, under the stimulus of the inflammatory cytokines produced by the infiltrate, produce growth factors. Among these factors there are the hepatocyte growth factor binding the CMET receptor, and the epithelial growth factor binding its EGFR receptor. Under these stimuli, the hepatocyte automatically starts producing the alpha-transforming growth factor binding the EGFR receptor as well. These three receptors stimulate the so-called delayed early response, where a number of genes, among which BCLX, p53 inhibitor, and MDM2 will be expressed. This will allow the cell to express high level of cyclin and therefore to enter the cell cycle. These processes will determine mitosis and increase in cell replication. In about 1 day, the mechanism is active, with peak replication activities, and in several weeks, the missing part of the liver is completely regenerated. However, at the end of this phenomenon, the supporting cells stop producing their own cytokines since they're now unnecessary, and, as a consequence, the hepatocytes return to the G1 phase. At this point, the lymphocytes produce the beta-transforming growth factor binding its SMAD receptor. The SMAD receptor stimulates the block of the replication gene expression, with the hepatocyte going back to G0 phase. There are also other phenomena of pathological hyperplasia, among which there is the benign endometrial hyperplasia. Benign endometrial hyperplasia is caused by an excessive hormonal stimulus. In case of benign endometrial hyperplasia, we can see the endometrium generating bigger, layered, and winding glands due to the increased number of cells. Another hormone-based form of pathological hyperplasia is that of the benign prostatic hyperplasia. Benign prostatic hyperplasia occurs in response to a part of the stroma during prostate aging, which would tend to cause prostate atrophy. This response, which is also known as embryonic reawakening, leads to prostatic epithelium proliferation. In the image on the right, there are two lumina of prostate glands. The one in the center, which is bigger and hyperplastic, has numerous cell layers. Remember that pathological hyperplasia is completely different from cancer, since it does not show any morphofunctional characteristics of the latter. However, it does represent a cancer risk factor in some cases. Finally, there are some forms of pathological hyperplasia that have nothing to do with hormones but are viral-based. This is the case of the human papilloma virus that, by infecting the squamous epithelial cells, can cause hyperplasia, commonly known as a wart. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.